All right, hello everyone. Welcome to our second general meeting. I hope you're all doing well on this election day and midterm season. If you can go to the next slide, please. All right, just some really quick reminders. You're allowed 2 excused absences per quarter, so great job being here today. Just notify your committee director at least 24 hours before the meeting. And again, we'll be using Slack for communication, and feel free to reach out to anyone else on board that's not your committee director if you have any questions or ideas for the year. And I will drop our presentation link in the chat right now. But you guys can access the roster here, which has like everyone's emails in the according committee. So if you want to reach out to someone, you can do so there. All right, and I'm going to turn it over to Samia and Arti, who will be doing a fabulous presentation on Parkinson's today.
Ooh, all right. So today we're going to be talking about Parkinson's, and the reason that we chose this as kind of our first content-based meeting is because we're probably going to be focusing on this disorder in particular a lot this year, and as well as like in like future quarters. So it's good to have like a foundation of like what Parkinson's is, and you know all the factors behind it. So first off, we're going to go over the biology behind Parkinson's, symptoms, and treatment. So the reason why Parkinson's is such an important disorder to focus on is because of the impact that it has on such a large amount of people. It's estimated that more than like 6 million people suffer from Parkinson's disease globally, though that number does range from 6 million to about the low 8 millions. And this makes Parkinson's the second most common neurodegenerative disorder, right after Alzheimer's. So it's believed that of the 6 million people globally who are believed to have Parkinson's, over 1 million are those who reside in the United States. Now, this means that nearly a sixth of the population suffering from Parkinson's comes from the United States, though that number can vary based on the level of reporting that various countries are providing about their health statistics. The most affected age group is 60 to 85 years, meaning that the most number of people diagnosed with Parkinson's are aged 60 to 85. However, there are other variations of Parkinson's, one of which is young onset Parkinson's disease, in which people can begin experiencing Parkinson's symptoms at an age as low as 20 and all the way up to 40 or 50 years of age. And of the 1 million people who have Parkinson's in the United States, about 2 to 10% of them have young onset Parkinson's disease. And finally, about 50 to 80% of people with Parkinson's can develop other neurological disorders such as dementia, and also including Alzheimer's, Huntington's, or progressive supranuclear palsy, which is basically the tendency to fall when someone is walking. And all of these disorders have symptoms that are very similar to those of Parkinson's, but they present either before or after the Parkinson's diagnosis. So here, we're going to watch a quick video explaining the biology behind Parkinson's and the symptoms that patients most commonly experience. Hopefully the sound works okay.
Parkinson's is the second most common neurodegenerative disease after Alzheimer's. The most visible features of this disease are a slowness of movement, muscle rigidity, and tremors at rest. There are also many, often debilitating non-motor symptoms, some of which may present before the motor problems. Parkinson's affects many areas of the nervous system and different types of neurons. However, much focus has been placed on neurons in brain regions associated with the overt motor symptoms, notably a region of the midbrain called the substantia nigra pars compacta. This region forms part of a major pathway in the brain that's critical for facilitating movement. In Parkinson's, dopaminergic neurons in the substantia nigra gradually die, leading to the malfunction of this pathway and the characteristic motor problems. Drugs that replace or mimic dopamine are often used to treat these motor deficits, but they grow less effective over time. Deep brain stimulation may also be used to treat symptoms. However, no current treatment slows the neurodegeneration. A distinctive pathology in most cases of Parkinson's is clumps of misfolded proteins within neurons. Lewy bodies are the most common type. characteristic component of these is a misfolded protein called alpha-synuclein. These molecules can form small repeated units called oligomers, or longer fibrils. Mounting evidence indicates that these are toxic to neurons and play a key role in driving Parkinson's. Unwanted proteins are normally cleared by the cell's different types of protein-degrading machinery. Some evidence points to these systems being overwhelmed by misfolded alpha-synuclein, and this might be a key factor in what kills the neurons. Parkinson's has also been linked to problems with mitochondria. Mitochondria provide cells with the energy to perform vital functions. They're highly dynamic and can fuse together or break up into smaller versions in response to a cell's energy demands. They can also be transported to areas of a cell that need them the most. However, in Parkinson's, these processes can be impaired, and mitochondria are unable to sustain proper neuronal function. As they become old or damaged, mitochondria are removed and replaced. Again, this recycling is thought to be disrupted in Parkinson's, leading to the accumulation of damaged or worn-out mitochondria. Another idea is that the cells called glia surrounding neurons may play a role in Parkinson's. As dopamine neurons are lost, one particular type of glial cell, called microglia, is thought to take up the resulting cellular debris, triggering an immune response. Once activated, they release inflammatory cytokines, which activate neighboring microglia and another type of glial cell called astrocytes. Chemicals released by activated microglia and astrocytes have been shown to injure neurons. It remains unclear which mechanisms drive the disease process in Parkinson's. What is clear is that with time, more areas of the nervous system develop pathology. And one emerging idea is that this happens through the transmission of misfolded alpha-synuclein. While the multifactorial nature of Parkinson's disease progression might make it hard to fully understand, it does provide a number of potential therapeutic targets for slowing or even halting the neurodegeneration. For example, enhancing the clearance of abnormal proteins and blocking their transmission, improving the function of mitochondria, and targeting the neuroinflammation are all currently active areas of research.
So as the video mentions, Parkinson's affects motor control mostly. So if you've seen any videos with Muhammad Ali or Michael J. Fox, or anyone with Parkinson's, you'll probably know that Parkinson's is usually associated with essential motor tremors. However, in order to make a full diagnosis, 4 factors need to be looked at in order for Parkinson's to be diagnosed, which include shaking or tremors, bradykinesia, which is also considered to be slow movement, stiffness in arms, legs, and or trunk, and balance issues. As well as physical impairments, Parkinson's is also associated with cognitive impairments. So you'll see slowed speech as well, loss of tone. There might be changes in writing. So Parkinson's, though, is a motor control disease. It also affects cognitive. It also leads to cognitive deficits. So as the video mentioned, there are 3 major cellular breakdowns that are associated with Parkinson's disease. So protein misfolding is a major issue. As they said, alpha-synuclein is a protein that is primarily found in neurons. When it forms misfolded oligomers, the protein-degraded machinery in the cell becomes overwhelmed as it needs to be able to get rid of all those misfolded proteins. As a result, a buildup forms which travels to other cells and leads to neuronal death, as the cells cannot cope with that many misfolded proteins. Another region of the cell that becomes impaired is mitochondrial breakdown. So the recycling of old and damaged mitochondria becomes disrupted and impaired. As a result, damaged mitochondria accumulates and initiates a positive feedback cycle in which mitochondrial dysfunction is promoted and cellular functions start to break down. Lastly, lastly, microglia release of cytokines is supposed to modulate neuron function in the cells of individuals with Parkinson's. It is seen that inflammatory cytokines are a major component. And so basically, normally microglia function as immune cells, but when the cytokines that are released are especially inflammatory, they can lead to the damage of other microglia and astrocytes. As a result, harmful chemicals are released and neuron death and injury occurs, which spreads across the brain. It is important to note that each of these cellular breakdowns are carried out via positive feedback cycles. So when one cell is damaged, that can lead to other cells being damaged as well. So there are three major areas of focus in treatment for Parkinson's, each relating to one of the cellular breakdowns that were discussed in the previous slide. So researchers are currently trying to understand protein degradation pathways associated with Parkinson's. As a result, they're focusing on finding the efficacy of clearance proteins in individuals with Parkinson's, as well as creating pharmaceutical treatments to treat alpha-synuclein buildup. If they do so, then they can decrease the amount of buildup and hopefully decrease the amount of neuronal death. Also, with regards to the mitochondria, researchers are in the process of determining whether or not mitochondrial dysfunction is the cause or effect of Parkinson's. So with this information, they will be better able to find pharmaceutical treatments that are most appropriate for treatment and basically determine whether or not they need to stop mitochondria from functioning improperly or maybe work backwards and find out what is causing the mitochondrial dysfunction. Lastly, researchers are focusing on the neuroinflammation caused by overactivity of microglia, as it is a hallmark in post-mortem brains of PD patients. It is important that researchers understand the mechanisms by which this occurs and how to treat it while continuing to keep the normal immune cell functions present in the brain. So this is a TED Talk in which the speaker discusses her experiences knowing people with Parkinson's and the current treatment options available.
How many people here know someone in their immediate circle who suffers from a neurodegenerative disease, such as Parkinson's, Alzheimer's, Huntington disease, or ALS? The show of hands I see before me is hardly surprising given what we know about these conditions. By 2050, more than 14 million Americans will be suffering from a neurodegenerative disease. To put this into perspective, that means for every 36 people you know, your friends, family, and loved ones, one of them will suffer from a fatal and debilitating condition for which there is no cure. In fact, every 65 seconds, one new Alzheimer's case is diagnosed. Every 10 minutes, one new Parkinson's case. And every 90 minutes, one new Huntington disease or ALS case. That means by the end of this talk, about 15 to 20 new cases of neurodegenerative disease will have been diagnosed. So what is a neurodegenerative disease? This is a condition in which nerve cells or neurons in the brain start to deteriorate. At first, these neurons begin to function abnormally, but over time, they're ultimately lost. And as more and more neurons in the brain are lost, symptoms begin to manifest, and they continue to worsen over time. Depending on the particular condition, whether it's Alzheimer's, Parkinson's, or Huntington disease, patients can experience a wide range of symptoms that can include memory problems, mood changes, or difficulty walking. At first, these symptoms might not have a huge effect on a patient's quality of life. If you imagine an Alzheimer's patient getting ready to go to work in the morning, she might not remember where she placed her keys, or she might get confused about what day of the week it is. Or if you imagine an early Parkinson's patient reaching for a cup of coffee, he might experience a slight tremor as he reaches for it, but ultimately, he'll still be able to pick up that cup and enjoy his morning coffee. But over time, as more and more critical neurons in the brain are lost, that same Alzheimer's patient may no longer remember her loved ones. And that Parkinson's patient, he might not be able to walk, to feed himself, and to carry out other tasks of daily living, tasks that we all take for granted. Eventually, both of these patients will require a full-time caregiver. And what do we know about the impact that this has on society? Well, like I mentioned earlier, by the end of this talk, about 15 to 20 new cases will have been diagnosed. And by 2050, the financial burden of just Alzheimer's disease in the US alone will be greater than $1 trillion. Neurodegenerative diseases can be caused by several different factors, including environmental and genetic factors. But ultimately, the number one risk factor is age. Due to current advances in modern medicine, fewer individuals are dying at young ages from preventable conditions such as heart disease, measles, and other infections. However, this means that the elderly population is rapidly climbing. Virtually every country in the world is experiencing growth in both the number and the proportion of elderly individuals in their populations. In the US, within the next 20 years, 1 in 5 Americans will be over the age of 65, and millions of these individuals will be suffering from a neurodegenerative disease. Unfortunately, the drugs that are currently available on the market only mask the symptoms of these conditions, and they don't actually get to the heart of the problem, which is the progressive loss of neurons in the brain. Those 2 patients I mentioned earlier, as they continue to lose more and more of their critical neurons, not only are their symptoms worsening, but these drugs are proving to be less and less effective at masking these symptoms. That Alzheimer's patient can't remember where she lives, she can't recognize her loved ones, let alone remember to take her drugs. And that Parkinson's patient, he's not able to stand up on his own, and he requires a walker to move around. At this stage of disease, both of these patients can no longer take care of themselves, and they rely on a full-time caregiver. As such, there is a glaring need for the development of a therapeutic approach that has the potential to stop or reverse the progression of neurodegenerative diseases. One of the most promising avenues for treating neurodegenerative disease are gene therapies. Each cell in our body contains DNA, which acts as a manual with instructions for cells on how to make different proteins. Gene therapy is a technique that uses pieces of DNA to treat or prevent disease. In the case of neurodegenerative disease, gene therapies can do one of 2 things. They can either introduce a new gene into a cell to allow that cell to make a beneficial or protective protein, or if a cell in the brain has a faulty gene that is causing neurons to die, gene therapy can actually turn off that gene or replace it. And this is revolutionary because it means that gene therapies can actually change the course of a condition. They can actually stop or prevent disease progression. However, one of the main obstacles standing in the way of developing gene therapies for treating these conditions is the blood-brain barrier. The blood-brain barrier is the body's way of protecting the brain from bacteria and other disease-causing agents. The downside of the blood-brain barrier is that it also prevents large drugs, including proteins and gene therapies, from getting into the brain when they're taken orally or when they're injected into the bloodstream. In most clinical trials for gene therapies, the drugs are directly injected into the brain. And not only is this extremely invasive, but when you consider that the majority of patients are sick and elderly, this is also quite risky. And furthermore, drugs that are injected into the brain in this way, they don't actually distribute well into the brain. They either remain at the injection site or they only penetrate the superficial layers of the brain. And this is problematic because it means that the drugs aren't getting to the deeper brain regions, which are usually the most affected in these conditions. All in all, I've painted a pretty daunting picture. I've basically stood here and told you all that in the next 20 to 30 years, millions of individuals will be suffering from a neurodegenerative disease, and that we currently don't have any adequate therapies available to treat these patients. Not only that, but even for approaches like gene therapies, which are really our best bet, we don't have a way to administer them to patients without resorting to invasive surgical procedures. But what if I told you that the key to treating a neurodegenerative disease, such as Parkinson's, could be as simple as a nasal spray? Intranasal delivery is a little known and underutilized approach for delivering drugs directly to the brain. And it's really as simple as it sounds. All it requires is administration of a drug into the nose. So how can this be? This is because there are 2 pathways that directly connect the nose to the brain. These pathways are a result of 2 main nerves that have their nerve endings inside the nose and that reach all the way up into the brain. They thus provide 2 channels that directly connect the nose to the brain. Once drugs are given in the nose, they can actually travel along the outside of these 2 nerves to sort of sneak their way into the brain. And once they're there, they then travel in the spaces that surround blood vessels, which allows them to be distributed throughout the whole brain. Intranasal administration has many advantages. It's very quick. It takes less than 1 hour for a drug that's given in the nose to reach the back of the brain. And it's non-invasive. All it would require is a simple nasal spray or nasal drops that a patient could administer in the comfort of their own home. And this also makes it easily repeatable. And by directly delivering drugs to the brain, this also prevents side effects in unaffected body organs because the drugs aren't really going to the rest of the body. One promising protein that has the potential to stop or reverse the progression of Parkinson's disease is called GDNF. GDNF protects the particular neurons, the dopamine neurons that die in Parkinson's. Most patients don't begin to develop symptoms of Parkinson's until they lose about 50 to 70% of their dopamine neurons. And these neurons play an important role in regulating movement. So it's really their loss that is causing the symptoms that are associated with the condition. GDNF can actually protect these neurons from dying, and it can make them healthier. And because patients don't begin to develop symptoms until they lose about 50 to 70% of their dopamine neurons, this means that there is a window of opportunity in which early intervention could allow these patients to live a relatively normal quality of life. So if that Parkinson's patient I mentioned earlier were to be treated with GDNF early on, before he developed debilitating symptoms, maybe he wouldn't reach the later stages of the condition where he couldn't walk or feed himself. However, GDNF is a large protein, and as such, it can't cross the blood-brain barrier when it's taken orally or when it's injected into the bloodstream. So what we really need in order to fully harness its therapeutic potential is a way of giving it to patients that is non-invasive. And this is exactly what we set out to do in the WASAC lab here at Northeastern. We thought that intranasal delivery was an ideal way of getting GDNF into the brain, but we wanted to take it a step further. Instead of just giving the GDNF protein intranasally, we collaborated with an industry partner to produce a GDNF gene therapy. And the idea was that this gene therapy would then be taken up into cells, and it would allow them to make their own GDNF, and that this would provide a continuous source of the protein in the brain. So we studied this approach in a rat model of Parkinson's disease, and we found that intranasal delivery of this GDNF gene therapy successfully bypassed the blood-brain barrier. Once it was taken up into the brain, it was taken up by cells which then became like mini factories, producing the protein all over the brain for at least 6 months. And what we found was great. The animals in our study went from losing 70 to 80% of their dopamine neurons to losing just 20%. And this was after a single dose. And they no longer showed any symptoms of the condition. Not only does this approach provide hope for the development of a therapeutic approach that may one day stop Parkinson's in its tracks, but it also serves as proof of concept for the development of other intranasal gene therapies for treating so many different neurodegenerative disorders. This approach has unlimited potential. There are numerous proteins that have therapeutic potential for treating each of these conditions. And if we could intranasally administer the genes that code for them, we might be able to stop the progression of these conditions. That means that in the future, if any of your friends, family, or loved ones are diagnosed with a neurodegenerative disease, it doesn't have to be a life sentence. As amazing as this is, there's still so much more that needs to be done to make this approach a reality for the millions of patients that are currently suffering. Studying intranasal delivery in depth and further developing potential gene therapies could be the key to curing so many fatal and debilitating conditions. I know better than to ask you all to go to graduate school to help us reach this goal. But I will ask that you consider helping nonprofit organizations and academic laboratories in any way that you can, and that you help fund research that's focused on overcoming the blood-brain barrier to treat neurodegenerative disease. If we could utilize this approach on a broader scale, gene therapies for all neurodegenerative disorders can and will become a reality, allowing millions of patients to find hope with just a simple nasal spray. Thank you.
Okay, so hopefully that was nice and informative. Maybe that gave you some ideas for research that you might want to do in the future. Who knows? Okay, so we're just going to do a quick recap of the video that we just watched, and that went basically over different treatments that are available to patients and each of their own pros and cons. So first off, we have oral drugs, and these include medications like levodopa, which basically enters the brain and is converted to the neurotransmitter dopamine. And then there's carbidopa, which mitigates the side effects like nausea, vomiting, and heart disturbances that come with taking levodopa. It's important to note that these medications don't cure or stop the progression of the disease entirely. They're only masking or lessening the symptoms of Parkinson's from progressing too quickly. And another con of these is that the medications often don't reach deeper portions of the brain where Parkinson's actually develops, which we learned about before was the substantia nigra. So overall, drugs can help make the disorder more bearable, but they aren't a definitive solution. Next off, we have gene therapy, and there's two types of these, one in which a new gene is introduced to begin the creation of proper proteins, and one in which a new gene is introduced to replace the faulty genes that are already existing. And this treatment option is usually kind of controversial because it's a very invasive surgical procedure, and it doesn't always produce results. And there's often aggressive immune responses from the body when new genes are introduced, which can lead to even more problems. These new genes often can't bypass the blood-brain barrier, which is the body's way of preventing infection from new genes and drugs, which the body thinks are foreign substances. So the body rejects them, and this leads to that immune response. And it can be quite risky for the patient in the long run. And finally, we have the treatment option, which our speaker talked about, and that's the intranasal gene therapy. And in this treatment, Parkinson's disease drugs are directly delivered to the brain via simple nasal spray and can generally bypass the blood-brain barrier, which we talked about earlier. And for the patient, this is much less invasive and much more convenient since they can administer treatment for themselves at their own home, so they don't have to go to the hospitals often. And there's also fewer recorded side effects, which is good, so that no problems are added on to existing symptoms. However, this therapy is very, very new still and still in clinical trials. The first published research about this therapy only came about 5 years ago in 2015. So we still don't know about how effective it'll be in the long run, but current research does find this treatment to be promising. Okay, so next off, we are going to split into breakout rooms. And we're going to have a discussion with some group mates about the various treatment options we heard about and which we think would be the most effective and the most ethical for patients. So we have surgical methods like deep brain stimulation. We also have gene therapy, medications and drugs. And one that hasn't really been talked about yet in the videos, which are non-invasive external therapies, such as the I Stop Shaking hand glove, which we would be focusing on this year. So we're going to go ahead and create breakout rooms. Let me stop share. Okay, I think our last few people are coming in. Do we have everybody?
One more group.
One more group?
One more group.
They're having a good discussion.
Great discussion going on. My goodness, we gotta ask them. Okay, I think they're coming back. We're good to go.
Okay, we good? Okay, awesome. All right, so hopefully you guys had good discussions with your groups. And let's continue talking. I just want to take a minute to talk about some research organizations, one of which is the International Parkinson and Movement Disorder Society. And this group basically focuses on talking about ongoing research related to movement disorders, specifically Parkinson's, and sharing their research through symposiums and seminars. And they also invite physicians to talk about their experiences treating movement disorders and the various approaches they take with Parkinson's patients. And if you're interested, this organization is also really great if you want to learn about movement disorders and Parkinson's in greater detail. And they actually have student membership for undergrad students interested in getting more involved. And by becoming a member, you can get access to their peer-reviewed journals, trainings, educational resources, lots of case studies, and a whole bunch of other stuff. So if you're interested in applying, we definitely do recommend becoming a member there. And finally, just really quick, earlier this year in September, they had a virtual congress in which they invited researchers, physicians, and other industry professionals to talk about their most recent advancements in the field. And they talked about new research, therapies that are being developed, clinical trials, patient cases. And recordings of these sessions could be available on the Movement Disorders Society website if you become a member, I believe, if you want to get a glimpse of some of the seminars that happened. It was just kind of interesting hearing about some of the most recent research happening.
So as we know that Parkinson's is the second most common neurodegenerative disease. So there's a lot of research going on both globally and at UCLA. So one research project that is going on right now is at the University of Cambridge, in which they're using stem cells to treat Parkinson's and seeing if that works. So in Dr. Barker's research, they're using stem cells to replace the depleted basal ganglia cells that make dopamine. And that dopamine is basically just a happy kind of neurotransmitter. So it kind of is part of the reward systems that are in place in the brain. So this potential treatment option is probably most similar to L-DOPA, which is a dopamine precursor, as it is able to mask the symptoms of PD, but it is not a complete cure of the disease. It's especially important to take into account that part of the PD pathology is the misfolded proteins, mitochondrial dysfunction, and neuroinflammation that has the potential to spread from cell to cell. So there might be issues when the new stem cells come in and are infected with the damaged cells as a result of contact. So we're just still trying to figure out whether or not this type of research will be good to completely treat and eradicate Parkinson's. So this is a video that just goes into that earlier video is just going more into detail. If you want to learn more, please look into that. Ongoing research at UCLA. There's so many labs at UCLA that work on Parkinson's. One of them is with Dr. Baton and Dr. Bronstein. And so they're part of a coalition of projects that are working to treat Parkinson's. And so fortunately for us, Dr. Bronstein will also be a guest speaker for us. So if you have any questions about his research, please do ask. And this particular research is looking at the inhibitory effects of the molecular tweezer CLR01, which could prevent the aggregation and misfolding of alpha-synuclein and therefore improve motor deficit. So it will be really interesting to have him in one of our lectures, and you can ask him more questions then about his research. So now we're going to be playing a quiz game. Hopefully it'll work well, but it's a bit like Kahoot, just not as pricey. So if everyone could just go on to quizwizard.com, we're going to send out I think the code that we need, and then you guys can log in to that using the code and then play our little quiz. And then I think we'll just be able to go over the quiz questions after we're all done.